We go at multiple places here and we record what species are there. So that's what we generally consider as providing a multivariate data set. Right? So what does it mean? That means that we have multiple responses. For multiple species, we measure, for example, if they're present or their abundance at each of these sites. If we, in R, that would give us a matrix where, that we see here on the slide, where we have the sites on the rows and species on the columns, and each of these cells contains the data. In contrast, multivariable means that we have multiple predictors. So measurements of the environment at each of the sites, such as temperature or soil moisture. And we can also have a separate matrix for that, where here we have the environmental variables or measurements on the y-axis, on the columns, and the sites again on the rows. And then each of these holds some kind of measurement. So that's just to clarify. So both our data, but also the model can be univariate or multivariate. But multivariate data can be analyzed with multivariate and univariate analysis. Well, univariate data, of course, only with univariate models. And multivariable data can be used in both. Where in multivariate analysis, we generally assume that these measurements are the same for all our species, all our responses. But of course, models can set parameters to 0 so that certain terms are excluded for certain species. Why do we want to analyze multivariate data? Well, it can give us some information about co-occurrence patterns between species instead of just occurrence patterns, essentially a species distribution, which is generally what we get with species distribution models or other analyses. And then we can start to infer why species co-occur, right? It can be, for example, due to them having similar environmental preferences and therefore occurring in the same place. Then also, of course, because they have similar history in the environment, such as dispersal and other limitations. And that might result in interactions, right? It doesn't have to be for two occurring species, co-occurring species. So essentially, we're looking at communities, right? Because multiple species then form community. For the record, the GitHub link is also here in the bottom of my slide for anyone that's looking for the material. And I'd suggest having a look there before we get to the next part with Jenny, so you can try and install the R package. So in this figure, I've just visualized the idea of an occurrence pattern where we have the abundance or occurrence on the y-axis and the environment on the x-axis. And then we can consider that the species has some kind of response curve, some kind of niche on the environment, where things of interest could be the tolerance of the species on the environment, and especially also its limits, right? Where it does occur and where can't it occur. And in contrast, we look at the second figure here, where we have now two species, the green and the blue one, where one is a bit wider response curve than the other. So one's a generalist, one's a specialist. And then we can also consider the space where they can both occur and what that means, right? So that's this dashed area here. And then we want to do it for a lot of species at the same time, right? So this is a very classical image where we have a bee, an ant, and a butterfly, and they both utilize these different flowers. But of course, because the bee and the ant and the butterfly, all three utilize this flower, can consider on how they can perhaps direct or indirectly affect each other by using that resource. This is another image of a community along an ecological gradient, where on the x-axis we have elevation, or actually moisture gradient at different elevations. And we see that this vegetation pattern changes in the environment, where at lower elevation we have this forest with low understory. And then as we go further along the gradient, the vegetation community changes up to where we at the highest elevation. We have lower trees, different kind of species, and different understory. So that's the idea of ecological gradient analysis, which is essentially an approach to study spatial patterns of species, right? That's what we're doing. Where our sites describe the environment, or again, we have this matrix of the predictors. where if we have two predictors that, for example, explain similar information, they can form a complex gradient, right? So for example, soil moisture and acidity that change along the elevational gradient. So an ecological gradient, ecological gradient represents the gradual change in the environment, such as temperature, while a complex ecological gradient represents change in several ecological gradients, right? So this idea of one variable that can explain two simultaneously. For example, yeah, near this, I already gave this example. So the idea is that we can represent that complex ecological gradient as a single factor or covariate, predictor, or latent variable, right? And these are all synonymous, and also an ordination axis, all synonymous terms. And from that follows the idea that gradients can be observed, or covariates, or it can be latent, so they can be missing. And if they're latent, they of course still produce the species patterns that we see. So we're still interested in determining what they are. I'm going to mention this because it's of relevance to the analysis later. Essentially, the ideas or studies have shown that few complex ecological gradients can often account for variation in species composition, which means that our community structure is generally few dimensions. So now I'm going to continue with ordination, which was turned by David Goodall in 1954, where he first applied factor analysis to an ecological community. Right, ordination is a bit older. Principal component analysis was developed by Pearson in beginning of the 1900s, 1904 perhaps. And the idea is that we're reducing the dimension of the data, where we have this, this data set of abundances that's generated by our underlying gradients, and we're trying to define a low dimensional space that explains our community structure as best as possible. You can also think of that as ordering species or samples along these ecological gradients that I've been talking about. Right, so examples are principal component analysis, which you can do with PR-comp function in R, correspondence analysis with the CCA function in vegan package, multidimensional scaling with principal coordinate analysis or NMDS in vegan, and then factor analysis, which is the factorial function in R, which is of importance here because that's kind of the precursor to generalized linear latent variable models. And ordination is most often inspected visually in a biplot. So biplot is essentially a tool to inspect ordinations, where this is an example of a biplot. We have arbitrary latent or not dimensions on the X and the Y axis. We have two species, again, the blue one and the green one, and we have sites 1 to 5, where distance between species and distance between sites indicates how similar species and sites are. So if species 2 would be on top of species 1 here, that means that we could expect them to co-occur in these dimensions. Class coordination methods have some issues. One of the ones is, for example, that ordination axis is often treated as fixed, as a parameter, while it is unobserved. So statistically, we want to actually treat it as random or mixed effect. We have a horseshoe or arch effect, which is notorious and familiar to a lot of ecologists, which is this kind of arch-looking distortion in ordination that's not necessarily believed to be of ecological importance or a result of the structure in our community. The downside is that these methods have assumptions that are very difficult or nearly impossible to check. And David Wharton and friends Huey published this paper indicating that not all ordination methods properly account for the properties of our data. So in general, these methods are not very flexible. So that's why here model-based thinking comes into the picture, where the idea is that we're applying regression concepts to multivariate analysis. And before that was difficult due to computational limitations, but of course, computers have been rapidly evolving. So now we're going to formulate an explicit statistical model That allows us to perform residual diagnostics, so check the assumptions of the model, perform model selection, and all other cool things that you can do with regression type analyses. Equation 1 is a multivariate generalized linear model where we have beta 0j being intercepts per species, so means, abundances, or occurrences. We have x or covariates, which are site-specific. And then we have the vector beta j, which are species-specific slopes, right? So this is a very basic model where we have a link function g that connects the conditional mean of a distribution that we choose to our linear predictor, which is here, this thing, right, with our intercepts and our fixed effects. And the idea is, of course, this is just a stacked species distribution model. So there's plenty of R packages that you can use to do this, but you can also just use the GLM function and fit that to multiple species at the same time. So it's a bit tedious. And you can, of course, only do it for observed gradients here. Making this a mixed effects model, we add a residual for all sites and all species, sites I and species J, which we assume to be normally distributed for sites, and we structure that with a covariance matrix that explains the connections between our species, the associations. So this is essentially a joint species distribution model in the flavor of Pollock et al., right? And what you fit in a lot of other packages, where we still have this link function g with our conditional mean, our species intercepts, fixed effects, and now we have this extra residual term here. And this is what I said. We have this per site. We assume a normal distribution around 0 and sigma covariance matrix. And this is just a standard mixed effects model. So you can actually fit this with LME4, GLMMTMB, or other mixed effects modeling packages like this, where this is then the syntax from LME4. You have your response variable here, abundance. This is just a vector of species names, your covariate x that has an interaction with species. And then you have this random intercept for species and a slope for sites. So this is a JSDM. That's all it is. The issue being that this model has an unconstrained variance covariance matrix, which has a lot of parameters, and the number of parameters increases very fast with the number of species. So we have a large number of species, we generally can't fit this. Either the software will be fussy and give you convergence problems, or you don't have enough data to estimate all the parameters. And that's where ordination comes into, model-based ordination comes into the picture, where now we're going to represent latent complex ecological gradients, but still with a model like in regression. And if you want to have a better or different introduction to this, or want to read about it, I can recommend this publication, Model-Based Approaches to Unconstrained Ordination from Francis Huey. And this means that we get all the benefits from regression, but also from ordination. So we can do Procastes analysis to compare ordinations, we can do biplots, we can still do model selection, residual diagnostics, still account for the properties of our data. We don't have to resort to using distance metrics, we can do hypothesis testing, and all other cool stuff. So generalized linear latent variable models, GOLVMs for short. It's the idea that we're adding a vector analytic structure to this variance covariance matrix of the random effect, right? And that's an ordination, because ordination means dimension reduction in this case. Where compared to the mixed effects model before, we're now decomposing this residual per site per species into a site-specific component vector of site scores and a species-specific component U and theta. And theta is a vector of species scores. So we're essentially assuming now that our residual per site follows a multivariate normal distribution where the covariance matrix is theta theta transpose. Benefit being this is a lot faster, and we have fewer parameters in the covariance matrix, then the number of parameters doesn't grow nearly as fast with the number of species. That also means that if you have more latent variables, you get these things. So latent variables are in these vectors of theta and u. You get better estimation of sigma. And if you have less latent variables, you get less good estimation of sigma. So it's all about capturing the proper dimension of sigma in this case. So it's still a mixed effects model. We have the number of latent variables, which are treated as random effects, unlike in classical ordination, where they're treated as fixed effects. And this gives us an ordination, right? With site scores, species scores, site scores U, and species scores or loadings theta, like in normal ordination. The main difference being that we don't have a rotation where the first latent variable is constrained to maximum variation. We're just going for best fit. And that produces plots like this, right? So this is, again, a biplot of species and sites. And the resulting model is equation 5, where now we have this linear, this conditional mean, conditional on the covariates and site scores, our intercept, our fixed effects, and our ordination with the latent variables or site scores U and species scores theta. Compared to some class coordination methods, principal components analysis is essentially a generalized linear latent variable model if you assume a normal distribution and if you treat the site scores as fixed effects. Correspondence analysis is an approximate generalized linear latent variable model with a Poisson or binomial distribution and with a row effect. Right, so a row effect is an additional site-specific intercept. Again, ordination acts as fixed effect. And the relation with NMDS is a bit more complicated, which I could provide in more detail. But what you need to know is that in practice, it produces similar ordinations. So GeoFEMs are more flexible than classical coordination methods, and they can also more easily be extended to include other things. So we get one method to rule all the other ones. Next, I think we have a short break, and I can answer some questions. And after that, Jenny will talk about the GeoFEM R package.